So starting with the recap, we talked about terminology because terminology is really important when it comes to genetics. Okay, some of the really important words in particular that I want to draw your attention to again are genotype versus phenotype. Okay, so when we talk about the genotype, we're talking about the genetics of the organism. Okay, when we talk about the phenotype, we're talking about the appearance. So another reminder, if you haven't got here in your course yet, then don't worry. Okay, this is like an extension. It's a hint of what is to come. If you've already done this, it's great revision. Okay, if you're here at the moment, then it's perfect timing. So, genotype and phenotype, genetics versus appearance. And they're not always identical. Our next one, I'll put my pen too low, would be dominant versus recessive. So here, we're talking about alleles. Okay, so alleles are different versions of genes, and we have dominant alleles and we have recessive alleles. So we're talking here about what is expressed. Okay, remember that a dominant is always a capital letter, a recessive is always a lowercase letter. Then, the other pair that we need to check in with before we get started is our homozygous and heterozygous. So this refers to which types of alleles we have. Are they the same alleles or are they different? So if we are homozygous, we have 2 copies of the same alleles. So we have either BB, homozygous dominant, or we have little b, homozygous recessive. And again, both of these are our genotype. For heterozygous, we have 1 copy of each gene. So 1 copy, of each allele. We always have 2 copies of each gene, 1 from the mum, 1 from dad. So here we will be big B, little b. So we are heterozygous. We have 1 dominant, 1 recessive. So then it's all about figuring out what alleles are linked to which genotype and therefore how we can get inheritance of different characteristics. So we can look at the phenotype, we can see what the characteristics are, and then we can work backwards often to get the genetics. And how we do that is our 2 little techniques that we mentioned last week is our Punnett squares and our pedigree diagrams. So what we're going to do now is just run through a few Punnett squares so that everybody can see how this starts to come together, how we can go from a parent to a gametes and then to different genotypes and offspring and how that becomes phenotype. Then we're going to move on to the fun stuff with pedigree diagrams that we didn't get to last week. Okay, so Punnett squares, we would have something like, we have a parent who is homozygous dominant, brown eyes. And we have another parent who is heterozygous. So we need to think about, okay, so based on that, what are our genotypes? So our genotype for homozygous dominant brown eyes, well, brown is dominant to blue, so we would have capital B, because we are homozygous dominant. Heterozygous, that's our genotype. So our phenotype is brown eyes. BB, capitals, is our genotype. Heterozygous means we would have one allele for brown eyes, one allele for blue eyes. Right, so if we turn this into a Punnett square, we're talking about what possibilities of genotypes and therefore phenotypes can we get from the offspring of these two parents. So we draw our box. Okay, and there we have parent 1 is BB. So their possible gametes are capital B, capital B. It's the same thing. Okay. Parent 2 is heterozygous. So here a possible gamete would be a dominant and the other possible gamete would be a recessive. So when we bring this all across now, in this square we would have top and left. So capital B, and the same thing if we move across here, we've got two capital B's because we're bringing this one from the row and this one from the column. If we take this bottom row first column is capital B, little b. Bottom row second column is also capital B, little b. So these are our possible offspring genotypes from these parents. So our genotypes we would like to know the genotype ratio. Okay, we're getting homozygous dominant to heterozygous, and we have 2 to 2. We never leave our ratio as an unsimplified. Think of your fractions. This will be 1 to 1. Okay, and then if we look at the phenotype ratio, okay, remember we said that capital B is brown eyes and it's dominant. So we have 2 dominant homozygous, they're going to have brown eyes, and we have 2 heterozygous with that dominant allele, so they'll also be brown eyes. So here we would have brown to blue, and our ratio is 4 to 0. All brown eyes, no blue eyes. Okay, let's say we have a different set of parents and see what happens there. So our next set of parents, we're going to say the parent 1 is homozygous recessive. In other words, they have blue eyes. Parent 2 is, again, let's have them, keep them heterozygous. So if we turn these into genotypes, our blue eyes homozygous recessive is little b, little b. Our heterozygous is the same. One of each, big B, little b. We can then do a Punnett square for this couple. Okay, so we have parent 1, little b, little b. Parent 2, big B, little b. Then we can fill in our possible gametes. We have a big B and a little b and a little b. So what do we get as possible offspring genotypes? First row, first column, we have big B, little b. First row, second column, big B, little b. Second row, first column, little b, little b. Second row, second column, little b, little b. So now, when we look at our offspring ratios, if you can spell offspring, it helps. Okay, so our genotype ratio is now heterozygous to recessive. But it's again 2 to 2, which is 1 to 1. Our phenotype ratio is a little bit more interesting. Because now when we look at brown to blue, brown would be these two that are heterozygous because they have the dominant allele. So that dominant allele is going to be expressed as brown eyes. So we would have two brown-eyed offspring, make myself space, and two blue-eyed offspring because they both have the recessive alleles. Okay, so that goes then to a 1:1. So we have the same genotype and phenotype ratio in this case. And what's important is that you actually work through a lot of Punnett squares. So when you get crosses in your textbook, when you get crosses in your lesson notes, make sure you work through those Punnets because it's an understanding in doing these Punnets that you start to see the patterns of when I cross this with that, what do I get? And that then actually helps you with the pedigree diagrams because the pedigree diagrams bring you back in, okay? As in, let's work backwards from our offspring, what are our possible parents? So if you understand the parents to offspring route really well, then it helps you with that offspring to parent route going back. Okay, so are we happy with Punnett squares? Give you a moment just to type through any questions quickly. Okay, what is recessive? So a recessive allele is one that is only expressed if the dominant is not expressed, not there. Okay, so if you have two dominant alleles, they will be expressed. If you have a dominant and a recessive allele, the dominant will be expressed. If you have a recessive and a recessive, then that will get expressed. So it's not that there's something wrong with it or anything like that. It's just that that allele doesn't get expressed if there is a dominant around. Does that make sense? Okay, cool. All right. So then we all sorted with homozygous, heterozygous, dominant, recessive, genotypes and phenotypes. So let's look at some pedigree diagrams. So pedigree diagrams look a bit different. Okay, in that case, we would have all of these things going on with different options of offspring, and then we would have traits colored in, and we've got to work backwards through them. So generally, you would indicate a square is used for a male, a circle is used for a female. Okay, and you've got to use your understanding of genetics and you've got to play detective. Right, so what we're going to do is we are going to work through some paper questions of these because while Punnett squares are very easy for me to draw off the top of my head, pedigree diagrams take a little bit more thought and planning. So let us take this to a different screen. Okay. So we should now be looking at a PDF. Let me just make it a size that I can see nicely with everything else. Okay, so we're going to do some practicing with pedigree diagrams. And this first one here, you see they haven't used squares and circles, they've actually used little mousies. So what we have is, they call it a chart, showing the inheritance of fur color in a small mammal that looks rather like a mouse. If the allele for white fur is dominant, which animal must be heterozygous for the gene controlling fur color? See how must is bolded. So it means that you got to take a note of that. It's important. Okay, right. So they tell us that the allele for white fur is dominant. So white, let's make him nice and spiky. The other one is black, and we'll make him nice and curly because my W's look rather too similar. So always be careful of things like that. You don't want to confuse yourself just with letters that don't look different enough. Okay, so we have our dominant, we have our recessive. Now we can start thinking about who's who. The easiest ones to do, of course, are your ones that are showing the phenotype for the recessive gene, because we know that this one is black. So he's homozygous recessive. So little w, little w. This one also, little w, and this one, little w, little w. And what is wonderful, we've already knocked out 2 options. We know that it is not D, and we know that it is not B, because those are both homozygous recessive. And we're looking, the detective work is aiming for the heterozygous one. Okay, so what else do we know about these mousies? We know that all these white ones are capital W. We don't know, because we don't know what that second allele is. Because it could be, remember, capital W, homozygous dominant, or it could be capital little, heterozygous. Now, what we want to find is which mouse must be the heterozygous one. So, this one could be heterozygous or not. We don't know, because this one could have a little w, or we could have a capital W. So we don't know what's going on here. When we get to this mouse here, which mouses are we looking at? Sorry, we're looking at mouse A in particular and mouse C in particular. So, mouse A and mouse B, our first 2 babies, had 4 babies. And all of these babies are white, which means that if you think about it, we had little w, and we can use our punnet here. Okay. All of these babies are white, so they've all got a capital W in them. Okay, which means when we add in our little w's, we needed a capital W and a capital W in both, which means that mouse A must be capital W in order to get our genotypes that we can use in this cross. That leaves us with mouse C, and when we looked at this cross that we did in the Punnett square, we agree that this one must be heterozygous. Okay, so mouse A is homozygous dominant, mouse B is homozygous recessive, therefore mouse C must be heterozygous. So our answer for this one is C. Okay, so that was a bit of a working through it, understanding what you've got, and then getting to your answer of mouse C. Are you happy? Okay, cool. Right. Let us do another question. Okay, so we love pictures with mice in, and it makes our life easier to work through. But we have different kinds of questions, and they like to do this with the multiple choice, is that they put it in words. So here we have white flowered pea plants crossed with red flowered pea plants. The offspring all have red flowers. If these F1 plants, remember F1 is our first generation of offspring. So we have parents, then F1, then F2, and so on. The next generation, the F2s, contains both red and white. Which statement explains this? So, we have a white flowered pea plant by a red. So, white by red. And in the F1, we have all of them white. And they're going by themselves, white. In the next one, F2, we get red and we get white. So, what does this mean in terms of what have we got? Well, as we just saw with the mouse, the only way to get all one color in our second generation, our F1 generation, is if they are all heterozygous. We saw that when we did our eye crossing as well. So, W, 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 W. We also know that white has to be our dominant because all of these offspring are white. So you must be capital, so dominant homozygous, and red must be lowercase. What have I done? I've got this back to front. Goodness gracious. Okay, ladies and gentlemen, obviously this Monday morning I am not reading very carefully. Okay, rewind, cut, restart. F1 all have red flowers. We are F1 pollinating themselves. Okay, all right, red times red. And we get white, F2, white, and red. Okay, so luckily with genetics, if you mix up your dominant and recessive, you can just back to front them. It's the same story. Only in this case, now we can see that our red is dominant. So we would have a capital R for dominant red. We usually use the letter for the dominant allele. So little r for white. Capital R, little r. So these guys are dominant and homozygous. These guys are recessive and homozygous. Okay, so, right. Now we can cross our red and our red, but they are both heterozygous. So, our red and our white. Our white one has to be little r again. Our red one can be homozygous dominant, or it can be heterozygous. So it's easier if we work through this before we then go and look at the answers. Because now we're sure, once we've read the question carefully, that we know that the allele for red is dominant. Okay, so the allele for red flowers is dominant. We like that one. And the and we like it there as well. We don't like the recessive. Okay, and then the F1 plants, we've said they are all heterozygous. So there we go. They are not homozygous. And our answer is A. Okay, so we can work through the cross ourselves. It's rather like a pedigree diagram because we're working from information rather than working from genetics. But it's the same skills of figuring out what are the possible genotypes that link to these phenotypes. Are you happy, or did I make some other silly mistake? Otherwise, we're going to do one more question quickly. So we have some happy people. Okay, right. So we've done mice, we have done flowers. Let us now do cats. So in alleles, in cats, the allele for short hair is dominant to the allele for long hair. So just to make life difficult, we have short hair, which is a big S, and long hair, which is a little s. A short-haired cat. Okay, so the short-haired cat. So the mother has to be dominant, but we don't know. She could be dominant and homozygous, or she could be heterozygous. So she gives birth to 5 kittens. 2 of them have long hair, which means that we have S we don't know, okay, and we have the little s, because these are our long hair kittens. So we need to think about now in what case could we get this homozygous recessive? And that's why I say it's so important to work through a lot of Punnett squares. Because you can think now, if you have a mother who is capital S, then that S is going to, that big S is going to come through in all of her offspring. So she can't be this one. The mother cannot be homozygous dominant, irrespective of what the father is. Okay, so the father, let's see if we need to figure out anything about him. We go to our options. First option says neither of the parents is heterozygous. Well, we can't get that. If we want to get this homozygous recessive, we're going to need either capital S, little s, mother against little. That's going to give us some fun and games with the father. If we get capital S, mother, heterozygous, and male who is heterozygous, then we start to also have a pattern. And so here we have our short-haired cats giving birth to long-haired kittens. So, one parent is homozygous. No, because we've said here the male can be heterozygous. So now we're left with two. The female is heterozygous or the male is heterozygous. Well, we've got here definitely female is heterozygous. But here the male could be heterozygous or not. So which answer is the correct one? You go back to the question and they say must be correct. The must one is the female cat is heterozygous. Because this is not necessarily for the male cat. Okay, so look for the clues in the question, work out your detectives, make sure you've done lots of practice with Punnets with different options, and then you're using that skill as a detective to figure out your genotype given your information on your phenotype. And that, ladies and gentlemen, takes us to the end of our half hour. I shall see you same place, same time next week. Cheers, guys.